All right, so we're going to pick up here talking about contraction of heart muscle. So the cardiac muscle cells are all interconnected by gap junctions and desmosomes. So you guys remember going back to
Week 1.
going back to like maybe week 2 we talked, or maybe week 1. I think we talked about tissues and where cells are interconnected by intercellular junctions, and we learned about how we have gap junctions, desmosomes, and tight junctions. Well, the gap junctions were the protein tubes that basically help connect these muscle cells together, and the desmosomes also help tightly hold these cells together. You might wonder, what the heck? Why would you want a lot of gap junctions between all of your cardiac muscle cells? Well, cardiac muscle cells are very short, unlike skeletal muscle cells, which could be like up to feet in length. A cardiac muscle cell is microscopically short. So that means that if you have electrical impulses from one cardiac muscle cell, they spread directly into the next through those gap junctions. So all of the cells of the heart are linked up by gap junctions, and we call this a syncytium, like a together cell. Basically, the heart functions as one big old cell, and electrically it's coupled. So electrical activities in one part of the heart will spread to another part of the heart through those gap junctions. Now, the aggregations of these gap junctions under a microscope look like what we call intercalated discs, which are basically the big old aggregations of gap junctions and desmosomes that help anchor these cardiac muscle cells together and allow for electrical currents to spread from one cell to the next. Okay. Now, one thing that's important to note, you guys, is that we do have specialized cell types in the heart that can spontaneously generate their own action potentials. Okay, we call these pacemaker cells. We'll come back to this later, but these pacemaker cells can just generate their own action potentials at a specific rhythm or frequency, and they do this independent of the nervous system. Okay, so if we zoom in on cardiac muscle, what we find is here's a cardiac muscle cell here, and it's linked up with other cardiac muscle cells. One cardiac muscle cell can branch into several different branches here. It's still one cell, though. We find that the place where these cardiac muscle cells connect, this is called the intercalated disc. These intercalated discs are full of gap junctions and desmosomes. So here are the desmosomes that hold these cells really tightly together. And then we also have gap junctions in here, which remember are the protein tubes. So why have a bunch of protein tubes connecting each cell together? You got it. Because the electrical impulse spreads from one cardiac muscle cell directly into the next through those protein tubes. Imagine remember as a kid, and you can go maybe to the play place, like at a fast food restaurant or something, and you can go from one little area to another through a tube. You might have friends in one side, and you're on this side. You guys can get to each other's sides through that tube. Well, imagine that is occurring in your heart, which means that the electrical activities in one cell can spread directly into another cell. This is important to note because what this means then is that once you get electrical impulses in the superior parts of the heart, like by the SA node, they spread directly across the atria, down into the ventricles, and then back up along the sides of the ventricles through those gap junctions. There are no synapses in the heart. You don't have neurotransmitters being sent onto the next cell to stimulate another action potential. This is a direct type of synapse. Remember the electrical synapse, where these cells were electrically coupled, and directly you can have molecules spread from one cell to the next through those gap junctions. Now, these gap junctions can be blocked. Conditions like acidosis can block these gap junctions, which then what that means then is you don't have electrical current spreading normally, so that can lead to arrhythmias or abnormal heart rhythms. So it's important to keep these gap junctions open. What's also kind of freaky, you guys, is that viruses can spread from one heart muscle cell to the next through those gap junctions, right? It's a tube. Why not? Well, if the virus particle is small enough, it could just diffuse straight through that little gap junction into the next cardiac muscle cell. So that viral infections in the heart can take advantage of our own gap junction system to really spread around a lot of the heart muscle, and that can lead to damage. Now, if you look on the inside of this cardiac muscle cell, we find a lot of the features that you saw in skeletal muscle cells. So we still have myofibrils, and the myofibrils were basically the big old bundles of protein filaments with the thick and thin filaments, and the aggregation of those thick and thin filaments from Z disc to Z disc was called a sarcomere. Okay, so we have one sarcomere, and what we find then is that these myofibrils have lots of sarcomeres that help produce contraction, or sort of a shortening of that muscle cell, which helps squeeze on the heart chambers and produce pressure that helps for blood to flow. But remember, the thick and thin filaments required two main things. We needed ATP for the myosin heads, and we needed calcium as well. If y'all remember going back to skeletal muscle, calcium we found in the sarcoplasmic reticulum. Remember that? So, like skeletal muscle, cardiac muscle also has a lot of SR, which is in blue here. All of this is sarcoplasmic reticulum. So when action potentials spread down the T-tubules, it activates the sarcoplasmic reticulum to release calcium. That calcium from the SR then flows on over towards the myofibril, where the calcium then can bind with troponin. Remember, troponin pulls on tropomyosin. The myosin head binds to actin, and that gets the whole sarcomere to shorten. But remember, each myofibril is made up of many sarcomeres together in sequence, so that you're seeing that the entire myofibril is shortening at the same time, which causes the entire muscle cell to shorten up. And that shortening of the muscle cell is that actually is the contraction itself. So we know that the heart's beating a lot, so you're going to see a lot of mitochondria to help supply enough ATP to keep this going. So our heart actually uses up a lot of energy too. And we find there's a nucleus here too. Now, heart muscle cells typically have 1 to 2 nuclei. That differs from skeletal muscle cells. Remember, they had many nuclei because skeletal muscle cells were much longer. You know, with a shorter cell, like a cardiac muscle cell, you don't need that many nuclei. Like 1 or 2 at the most is fine. But these cells are also more highly branched. So if you look at this under the microscope, you can see it looks a little bit more irregular. So here's one cardiac muscle cell. You can see that between its edges, there's intercalated discs. So what do you find in these intercalated discs? Nice. Gap junctions and desmosomes. And then which one is for support? Desmosomes. And then the gap junctions were for communication. So electrical current then can spread from one cell directly into the next. And it's pretty fast. It's pretty fast. Not too fast, but fast enough to make sure you have a quick heartbeat. So these are some differences, you guys, between skeletal muscle. First of all, cardiac muscle cells have automaticity, or they're autorhythmic, which means they can generate their own action potentials. That differed from skeletal muscle because we needed to stimulate skeletal muscle to induce a contraction. You didn't have skeletal muscle that could contract on its own. It had to be stimulated by the nervous system. That differs from the heart because about 1% or so of your heart muscle cells are autorhythmic, which means they can generate their own action potential independent of the nervous system, like spontaneously. And they do it at a certain rhythm. Earlier we talked about how the SA node generates action potentials at 100 times per minute. Well, that's autorhythmic. So at a rhythm of 100 times per minute, you're generating action potentials, right? And then there's other parts that can generate them at 60, 40, 20, and we'll go over those, okay? Now, they don't need the nervous system to be stimulated, and they can depolarize the entire heart. So when we talk about these pacemakers, like the SA node or the AV node, once they generate action potentials, those action potentials spread across the heart, and it depolarizes, or basically initiates action potentials in all of the heart areas, okay? Not at the same time, but in a specific order, and that's dependent on a conducting system, which we'll come back to, okay? Now, this differs from skeletal muscle because skeletal muscles can't generate their own action potential spontaneously. You don't have action potentials generating spontaneously in a skeletal muscle, like intentionally. Sometimes it can happen accidentally, but this differs from the heart because your heart does not need the nervous system to operate. This is why you can see that heart can beat outside the body. You know, that can happen actually. Because you don't need a nervous system to stimulate it. You know, whereas skeletal muscle won't just start contracting unless you're stimulating it somehow. Now, all cardiomyocytes contract as a unit. They're all or none. So it means once you get some of these cardiac muscle cells to contract, because they're all linked up by gap junctions, that causes the next nearby ones to contract, and the next nearby ones to contract, right? So it's kind of like telephone, you know, like if we all played telephone in here, and we all just start kind of passing a message on to each other, you know, if I initiated this stimulus, eventually anyone in the back will also, you know, get that signal, right? It could take a little bit of time to get there, but we're all going to kind of communicate, you know, as one, if that makes sense. Now, what we do find, though, and you guys, is in cardiac muscle cells, the refractory period's longer. If y'all remember going back to the normal action potential, like in neurons or skeletal muscle, The action potential itself was like milliseconds, like 1 to 3 milliseconds. What you're talking about is 1/1000th of a second or 3/1000ths of a second. So 3/330ths of a second. So it's really, really short. That differs from the cardiac action potential, because their action potentials are about 1/4 of a second in length. So 250 milliseconds, 1/4 of a second or so. That's how long these are. So they're much longer action potentials in a cardiac muscle cell than skeletal muscle. Now, by having a longer action potential, what that allows for then is you ensure that the heart muscle doesn't just flutter, or doesn't just kind of like really kind of quiver, right? Instead, you get a nice smooth contraction, which pumps that blood, and then you get a nice relaxation before the next action potential begins. If the action potentials could be simultaneously, like side by side, the cardiac muscle would just kind of quiver, and that's not going to pump blood very well, right? You want to have a nice smooth contraction, which is mediated in part by a longer action potential, also a longer absolute refractory period, which prevents tetany. This differs from skeletal muscle because in skeletal muscle, you can have not action potentials overlap, but tension can overlap. And you get skeletal muscle to a maximal tension by having tetanus, which is where you have summation or addition of tension. That differs in skeletal muscle because you don't want tetany in skeletal muscle. You do not want a sustained contraction in cardiac muscle, You don't want a sustained contraction in cardiac muscle because what would happen if your cardiac muscle just contracted and then stayed contracted? Yeah, you got to relax to fill, right? So with cardiac muscle, contraction and relaxation are equally important. You need to contract to pump that blood, but then you need to have that followed by a nice relaxation to fill the heart chamber back in with blood. And what allows for that is this longer absolute refractory period, which ensures not only that you get a nice, clean and smooth long contraction to pump that blood, but it also ensures that you get a nice relaxation before the next action potential begins. Now, what's important to note, you guys, is that because this refractory period is long, like 1/4 of a second in length, that means you can only have a certain maximal level of heart rate. It's in the hundreds of beats per minute, but you can't go any faster than that because you can't generate another action potential soon enough because there's a refractory period. Okay, that's the period of time where you can't generate another action potential. It's longer here, and it makes sense with respect to the heart because you want the heart to function as a pump, not to function as a lifting or pushing force, if that makes sense. So you need to have a nice long refractory period to contract and then relax. That you can fill it back up with blood, then contract and relax, right? Or remember the arm thing. Contraction and relaxation. So contraction, relaxation, contraction. Right? That way they're separate events. Because what if we didn't have a long refractory period? You could have it just gets all crazy. So So earlier we talked about how the heart has a conducting system. And this conducting system means that there are specific cells in the heart, remember about 1% or so of heart muscle cells, that they don't produce a contraction. Their sole purpose is to generate action potentials and spread those action potentials. So this conducting system of the heart doesn't participate in the muscle contraction itself. It just helps spread the signal. Okay? And it all begins with the SA node. The SA node, or sinoatrial node, it's found at the juncture of the superior vena cava and the right atrium, right at that juncture. That's the name sinoatrial node. And this is the major pacemaker of your heart. And the reason why we call the SA node the pacemaker of your heart is it generates action potentials at about 100 times per minute. And because it's the fastest at generating action potentials, it sets the pace for the rest of the heart. Okay, that's the SA node. It's the pacemaker of your heart. Now, is a normal resting heart rate 100 times per minute? No. So what's going on here? Well, this is true 100 times per minute in absence of any kind of nervous system input. But we know that your heart does have nervous system input. It gets vagus nerve input. It gets sympathetic nerve input. And that can change your resting heart rate. But if you cut those nerves, then the heart would beat at 100 times per minute. Give or take some. But that's the general average range, which is the pace or the rate at which the SA node or sinoatrial node fires action potentials. That's dependent on different genetic factors that I don't want to get into, but if you're interested, we can talk about them later. Now, The SA node is connected to another node of this pathway, guys, called the AV node. AV node is the atrioventricular node because it's found right at the juncture of the atria and ventricles, right in the center part, kind of right by the interatrial septum and interventricular septum, right where they meet. You have the AV node there. Now the SA node is linked up with the AV node through what we call internodal pathways. So that when action potentials from the SA node begin, they spread across the atria through those internodal pathways to the AV node. So that means the atria are stimulated first, which means they contract first. And that makes sense because they're going to pump blood down the ventricles. Now the AV node fires action potentials around 60 times per minute. So even if you didn't have an SA node, your AV node could still generate action potentials at about 60 times per minute. Okay? That's in the absence of any kind of nervous system input. So that way you have a backup plan. There's two different nodes here. One fires faster though. And the reason why we talk about the SA node or sinoatrial node as being the pacemaker is because it fires at the fastest frequency, it's setting the pace, right? Like let's say if it's in the back and it's pushing everyone at the same pace, right? If they might want to go slower, but if they're being stimulated, they can't go any slower. Because the SA node is the fastest, it's setting the pace. If that makes sense. Think of the SA node as that really impatient driver behind in traffic who's like, go, hurry up, go, you slowpokes, right? But then the people actually do start going faster, right? You're probably not saying slowpokes if you're that mad, right? You're saying other things that I don't want to be recorded, right? You slowpokes, you tortoises, I got places to be. So the AV node, you guys, is what links up with the AV bundle or bundle of His. Now the AV bundle of His, think of this as an anatomical bottleneck. It's actually a place where action potentials slow down. You might wonder, why the heck would you want action potentials to be delayed at the AV bundle? Well, remember, we just had action potentials come from the atria, which caused the atria to contract. Then the action potentials, are near the AV bundle. Why wouldn't you want the action potentials to just go straight to the ventricles?
Because have a delay and so they can control rest.
Good. You want to have enough time for the atria to contract to allow for adequate filling of the ventricles before the ventricles start contracting. And so it's the AV bundle or bundle of His that functions as a bottleneck. It slows things down. And actually, if you look at the anatomy of this, you guys, it looks like a bottleneck. It's basically a place where lots of these fibers, the connecting network, they all kind of converge, and then it's like 5 lanes of traffic that go to one, right? If you have 5 lanes of traffic trying to merge into one lane, you're definitely going to slow down there. Okay, but after you get past that slowdown, it opens up and you start going fast again, right? So think of the AV bundle as this anatomical bottleneck. It temporarily slows down the action potential propagation. That way, it allows enough time for the atria to contract before the ventricles start contracting. The AV bundle, or bundle of His, it's found right at the superior part of the interventricular septum. It divides into the right and left bundle branches. The right bundle branch goes to the right ventricle. The left bundle branch goes to the left ventricle. Okay? Now, those eventually branch into what we call the subendocardial conducting plexus, or the Purkinje fibers. Same thing. So, these Purkinje fibers are the fibers that directly stimulate the ventricular muscle. You don't get ventricular muscle stimulation from the AV bundle or the bundle branches. And that makes sense because if the AV bundle is more superior and the bundle branches are also kind of more superior, you don't want the superior parts of the ventricles contracting first. Remember we talked about earlier, you want the apex to contract first, then have the superior parts of the ventricles contract. That way, your ventricles are actually forcing that blood from the bottom up and out of the ventricle. And so you don't get ventricular muscle contraction until those action potentials get to the Purkinje fibers. Once at the Purkinje fibers, that's what these connect to the muscle cells, which then generates contraction in the ventricles. Okay. Now, even the Purkinje fibers can generate action potentials, and they generate action potentials around 30 per minute or so. So even if you didn't have an SA node, or an AV node, or an AV bundle, or even bundle branches, if you only had Purkinje fibers, your heart could still potentially beat on its own. It can still beat at 30 times per minute. Yes. Not in the long term. Yeah, not in the long term. Temporarily, yes. You know, and this degree of bradycardia is pretty low. Unless you had really good cardiac output, which we'll come back to later, you probably couldn't support your body's tissues very well at 30 beats per minute, right? And, unless you're a trained athlete. Yes, and then if someone can basically shock your myocardium, you can reset these pacemakers.
I have a question.
Yeah.
So, if the SA node beats at 100 beats per minute, how do you not, like, and the ventricles are beating at 60 or whatever it was, beats, how do you not have, like, 2 beats for every ventricle beat?
Excellent question. So the question was, if the SA node's firing at 100 beats per minute, and the ventricles will fire at 30 beats per minute, how do you not have multiple atria contractions before you have ventricle contractions? That's an excellent question.
Because the SA is controlling everybody else.
You got it.
Remember,
It's the pace.
driver that's honking the horn.
Right, but that's what fires the atrium?
Well,
Or is it the AV node that fires the atrium?
let me clarify this really quick. So this would be the beats per minute in absence of other
Right.
impulses. So if you could somehow not have the SA node or the AV node fire at all, ever, then the ventricles would still contract at 30 per minute. But because we have an SA node that can fire much faster, it's setting the pace. It's pushing everyone faster. So it's not the case that your ventricles would only beat 30 times per minute, because they could be stimulated by the faster SA node. So the SA node set the pace.
In the absence of
I think
Oh,
she's
she's
asking.
wondering
Well,
why-
I'm just wondering
what
because
is-
it's 100, but your heart actually only beats like 75.
Good, we'll come back to that. So-
That's because the bundle of discs force, the
We'll come back.
bottleneck
No, that's not it. That's not it. We'll come back to that though. You're right. So let's go through this pathway, you guys. So we got the SA node up here, where the right atrium meets the superior vena cava. You got the AV node right over here, kind of near the juncture of the atrium and ventricles. The SA node is linked up to the AV node by the internodal pathways. You can't say that without sounding like a huge nerd, right? Like internodal. Internodal pathways. But I embrace it. I embrace it. Why not? So SA node links up with the AV node by the internodal pathways. So here's the AV node. After the AV node, we got the AV bundle or bundle of His. That bundle of His branches into the right and left bundle branches, which then go by the apex. And then they have little fibers that come off called the Purkinje fibers, which start stimulating the ventricular myocardium from the apex up, which means that in the sequence of cardiac events, we find then if action potentials begin in the SA node, and then they travel the AV node through the internodal pathways, the atria will become activated or depolarized first, which is what we're showing in green here, you guys. So atrial depolarization happens first. Then what happens is that once those action potentials get to the AV bundle of His, there's a slight delay, which allows for time for the atria to force blood down in the ventricles. Then those action potentials travel down the bundle branches to the right and to the left, and then they start activating or depolarizing the myocardium from the apex up. And what this allows for then is the myocardium can start to contract from the apex, and so that blood's actually pushed up and then out through one of the semilunar valves, whether it's the aortic semilunar valve or the pulmonary semilunar valve, which isn't shown here. And so we actually can measure this on an ECG or EKG. It means the same thing, you guys. ECG is electrocardiogram, where cardio is spelled with a C. EKG is electrocardiogram, where cardio is spelled with a K. That's the German form, but we still use the same things interchangeably. So ECG, EKG, they mean the same thing. Just one's German, one's English. I like EKG. It rolls off my tongue more easily. So I say EKG, I mean ECG, same thing. So what we find then, you guys, is that an ECG or EKG measures changes in the electromagnetic fields of your body over time. So you get time here, and this is the amplitude of these changes in electromagnetic fields. We can call this voltage. I don't want to confuse y'all, but just think of like changes in electricity. Okay. So what we find then is that there's these specific and deliberate deflections here in electromagnetic activity, right? So what we find is there's a little wave here, followed by a bigger wave, and then another wave. So this is actually showing the electrical events of the heart over time. Now, what's nice with these EKG strips, you guys, is that time is standardized. So that every small little square here is 0.04 seconds, which is 40 milliseconds, okay? Every large square, right, all of these big squares, is 0.2 seconds, which is 200 milliseconds. But remember, if 200 milliseconds is 1/5 of a second, then you only need 5 of those big squares for 1 second, right? So if you look at this, you got 1 big square, 2 big squares, 3, 4, and about 5, right? So if you got 5 big squares here, that means that between each beat, that's 1 second. So if you have a beat every second, how many beats per minute is that? 60. You're right. Good. In fact, there's another really easy way to calculate heart rate based on this, you guys. You can just count the number of big squares between each of these peaks and take 300 divided by the number of those big squares. So if you see 1, 2, 3, 4, 5, what's 300 divided by 5? 60, right? And it's about 5. It's not right on 5. You guys see how this line's a little bit off? Like if it were right on that line, it would be right on 5. But it gives us a really kind of a close approximation. You know, it means like we're not precise, but we're close. We get the gist of what their heart rate could be. That only works if they have a normal rhythm, right? If this interval here is varying, then you can't calculate it just by looking at one interval. So then it gets a little more complicated there. But if someone has a normal rhythm, like a normal interval between heartbeats, then what you can do is you just have a nice quick way to figure out heart rate. Okay, and that's sort of what you do like in a more of an emergency situation. If you don't have time to like sit there and like count the number of beats in a minute, right? So what we want to talk about too, guys, are these different waves. So what we have is a P, QRS, T wave. Okay, the P wave is this small one here. It's associated with atrial depolarization, and it makes sense how that happens first, right? Because what's the first? Where's the first action potentials begin? The SA node, right? Which is near the atria. So it makes sense you'd see atrial depolarization first. Depolarization means action potentials are beginning, right? Followed by QRS. QRS wave is ventricular depolarization. Why does it make sense that this would come next? Because the action potentials have to get there, right? So the action potentials eventually get to the ventricles. You guys see how long it takes? Not very long. That's like 120 milliseconds. So it's fairly fast, but it's slow enough to allow for these mechanical events to work normally. This is not measuring mechanical events, by the way. EKGs or ECGs only measure the electrical events of the heart. That's an important distinction because this is only telling us what's going on in the heart electrically, not mechanically. But we do know that electrical events can stimulate mechanical events because once these action potentials are, in a particular area, they should generate contraction. So QRS wave here correlates with ventricular depolarization. This means the ventricles are now starting to have action potentials present. And then you have a longer delay followed by a big old T wave here. So you guys notice in alphabetical order, P, QRS, T. And then the T wave is ventricular repolarization. Remember going back to action potentials, this is where action potentials are kind of repolarizing back down to a resting voltage. So where's atrial repolarization then?
It's happening during the ventricle depolarization.
Yeah, it's in the QRS wave. So we had atrial depolarization. Atrial depolarization was the P wave. QRS wave was ventricular depolarization. T wave was ventricular repolarization. But where's atrial repolarization? It's in the QRS. In fact, the shape of the QRS is actually in part due to the repolarization of the atria. But it's in there. That's why we only have 3 waves for 4 events, right? Because 2 of those waves, or 2 of the events, share the same wave, which is the QRS wave. That QRS wave represents atrial depolarization, where the action potentials are going away, as well as repolarization and ventricular depolarization, when the action potentials are beginning in the ventricles. Why is ventricular depolarization a much larger wave than atrial depolarization? Way more muscle in the ventricles, right? Think about how thin the atrial muscle is. It's super thin, several millimeters thick. ventricular muscle can be as thick as about an inch, especially the left ventricle. Because you have way more ventricular muscle, its signature on the EKG is going to be much larger. Okay, so it should always occur in this order: P, QRS, T. P, QRS, T. And this is a normal ECG here, guys. This isn't like a disease process. So we find that a normal sinus rhythm is between 60 and 100 beats per minute. So if someone has a resting heart rate between 60 and 100, then we still consider that normal. If it's less than 60, what do we call that? Bradycardia. And if it's more than 100, what do we call that? Tachycardia. Good. Because if it's less than 60, now you're going lower than the intrinsic firing rate of your AV node. And if you're going more than 100, then you're going faster than the intrinsic firing rate of your SA node. So you're outside of normal limits there. Okay. So as long as you're within this range, we still consider that normal. You might find, though, in some individuals, you can find that their resting heart rates may be greater than 100. Doesn't always mean that's a disease process. Or like in some athletes, their resting heart rates can be much lower than 60, right? Like I guess like, you know, people do very aerobic activities like cycling or sprinting. You know, their resting heart rates can be like in the 40s, which basically means they can pump out the same amount of blood in less beats, which is pretty phenomenal, which basically means their heart is stronger, right? If it takes 100 beats or more to pump out the same amount of blood as it takes somebody else to 45 beats to do, you know, what that suggests then is either the heart's not filling very well or it's not contracting very well.
Her resting heart rate is really low. It's like 40 when we took it in lab.
Nice.
Yeah, like 42.
So do you exercise? Yeah.
Every day.
Cool. That makes
A lot of
sense.
aerobics.
Yeah.
Every time I go to the doctor, they're like, "Is this normal?"
Yeah.
Yes.
Yeah, because that's really low.
Yeah, it is. They're like, "Are you okay?" I'm like, "Yeah."
That just means you have a really strong heart, and it's really efficient at pumping out an even greater amount of volume of blood in one beat. So it takes less beats to get enough blood to supply your body's tissue with oxygen. Well, it's healthier. Yeah. I mean, it's not always cut and dry answer like that, but it is healthier to have a slower heart rate. You may have heard before that people have a finite number of heartbeats in a lifetime, and there's some data that suggests that, but it's not good. It's not good enough data to say that whether or not that's true or not. You know, I've heard somewhere it's like so many million heartbeats you have in a life, and if you get to that point, then you'll die sooner. I don't think that's necessarily true. That's my opinion, but I think you need more data to figure that out. Yeah.
So why do babies have a higher heart rate
I mean, it could be a variety of factors. It could be that their hearts aren't, strong yet, so it takes more beats to get enough blood to supply their body's tissues.
than?
It
I'll
could-
take that.
Okay, but I was thinking it could be a lot of things. It could also be too that maybe because they're growing and their tissues are very metabolically active,
They might need more blood.
they might need more blood flow. Exactly, you got it. So what's the P wave represent, you guys? Atrial depolarization. QRS, ventricular depolarization, and T wave, ventricular repolarization. Now, what also happens during the QRS? Atrial repolarization. Good. And it's not very significant though. Like if you could somehow separate ventricular depolarization and atrial repolarization, you can't really even see atrial repolarization. Like if the atria start functioning independently, you're not going to see like an atrial repolarization wave anyways, because it doesn't have that much of a signature. So it's more important to say that the QRS wave is mostly due to ventricular depolarization. Yeah.
Are you gonna ask us about repolarization?
You want to know that it occurs during the QRS wave. Now that's important to know because it's part of the cardiac cycle. And you got to know that the atria are repolarizing because they have to reset, right? And repolarization is when you're going back down to zero, like near resting voltage. Not zero, but a resting voltage. Absolute refractory period occurs during the action potential for the cardiac action potential. And that cardiac action potential is about 250 milliseconds long. So because depolarization is part of the action potential, then yes, depolarization is still part of the absolute refractory period. So this is showing a typical ECG recording, guys. So what's our P wave represent again? Good, atrial depolarization. Then QRS, ventricular depolarization, and the T wave here. Ventricular repolarization, good. And then what's this little guy right here? What the heck? Sometimes there's a U wave. And the U wave you can find with bradycardia, like you might have a U wave if your heart rate's in like 41. Or under conditions of hyperkalemia, which means high blood potassium. Okay, and sometimes there's an additional wave there. It can also be normal in children, but it should disappear by age 40. If it exists in someone after age 40, it may indicate an underlying pathology. But you guys know notice there's intervals here? Like there's a PR interval, QRS interval, ST segment, and QT interval. These intervals are important to note because what they represent then is the speed of conduction, right? Knowing that there's a specific PR interval, this basically tells you how long it takes action potentials to go from the atria and stimulate depolarization of the ventricles. What do you guys think it would mean if this PR interval were much longer? It's taking longer for action potentials to stimulate the ventricles, and that may indicate heart block. Heart block is a condition where action potentials either take longer to get to other heart areas or they never get there. So if you see longer PR intervals, that could be a heart block. So and there's different types, so we'll get to later, right? What about if this QT interval is longer? The QT interval is longer. What does that tell us? You got it. It's taking longer for the ventricles to have their action potential depolarize, followed by repolarization, which does correlate with contraction and relaxation. And that may indicate an underlying pathology. So it's important to note that these intervals exist. Don't worry about the intervals today. Just want to know the activities of these at these different waves. And what does an ECG tell us?
Electrical impulses of the heart.
Yeah, electrical impulses of the heart. Is it one action potential? No, it's millions or even billions of action potentials that you're finding in all of the cardiac muscle cells at the same time, right? This is not one action potential. So that's important to know. And don't worry about the direction of these waveforms, you guys, like different deflections. They don't mean much when you're only looking at one reading here. Okay? Now, if you take more advanced EKG classes and you can get different leads, different readings, these deflections tell you about precisely where action potentials are spreading with respect to those leads. You know, if you're comparing a lead like that's on your hand to your foot, and you're looking at the deflection of these waves with that relationship, it can tell you precisely which direction these action potentials are flowing in the heart. And that may be important to know if someone has like an arrhythmia. And that could be due to like a blocked blood vessel, which means that parts of the heart aren't conducting action potentials well. And you can actually pinpoint precisely in the heart, based on EKG readings, like where different blocks might be, which is actually pretty cool. But don't worry about that for now. So, what does a P wave represent again? Depolarization. How about T wave? Ventricular repolarization, and then the QRS? Ventricular depolarization. A little bit of atrial repolarization. Good. So what this slide shows you guys are basically those electrical activities in the heart as they relate to the EKG. Now, this is important to note with the cardiac cycle. If you know what the P wave represents on a cardiac cycle, or on EKG, I'm sorry, then you can guess what mechanical event would happen next, right? So if the P wave represents atrial depolarization, that means the action potentials are starting to begin, and they're spreading across the atria. What happens after the action potentials spread across those atria? Then the atria contract. So what you should expect then is after a P wave, shortly after, you should expect atrial contraction. That's not what these data are showing us, but you should expect that to occur. Okay? What happens next? Well, we get a delay. Why is there a delay between P and QRS waves? Good. There's a delay because you want time for the atria to effectively pump that blood in the ventricles. But how does this occur anatomically? Like what's the anatomical correlate of that delay? You got it. The AV bundle of His. So what if someone had a bundle block? What should you expect? How about a partial bundle block? Like not completely blocked, just partially blocked.
It would delay as much.
Yeah, you might have a longer delay. Yeah, a longer delay. So you might have a longer PR interval. And then what happens next, you guys? The action potential spread across the atria through those internal pathways. They get to the AV node. As the action potentials are spreading down the interventricular septum and in the ventricular muscle, do you guys notice how this turned red up here? Red here indicates atrial repolarization, which correlates with ventricular depolarization, which both occur during the QRS wave. So here it is. And then what follows next, you guys? Well, the atria are done repolarizing, while the ventricles are still depolarized. And then, then the ventricles start to repolarize. And what does that correlate with? T wave, which is here. You got it. So what's really cool about the EKG is just by looking at these different waveforms, you're basically seeing the electrical activities of the heart in real time. So that's pretty cool.